Principle of agarose gel electrophoresis. Agarose gel electrophoresis is a fundamental molecular biology technique used to separate macromolecules, particularly DNA fragments and RNA, based on their size and charge by passing them through an agarose gel matrix using electric field. Here we are performing this experiment to separate the macromolecules such as DNA and RNA fragments based on their size and charge by using an electric supply in an agarose gel matrix. What are the requirements to perform this experiment is an electrophoresis chamber, gel casting trays, sample combs, electrophoresis buffer, loading buffer, staining, power supply and transilluminator. This is the horizontal electrophoresis chamber and gel casting tray, sample combs. This is the final setup. Here power supply electric field is supplied. This is the horizontal electrophoresis chamber. First we have to perform this experiment by preparing a gel preparation. The gel is prepared by dissolving the agarose powder in an appropriate buffer such as TAE and TBE to be used in electrophoresis. The agarose is dispersed in a buffer and heated. The melted agarose is allowed to cool sufficiently before pouring the solution into casting tray. First we have to take appropriate amount of agarose. Let me take as 1 gram dissolving 100 ml of buffer. Buffer we are using here is TAE. Full form. What is the full form of TAE? Triacetate EDTA. EDTA means? And another one is TBE. We usually prefer TAE because in TBA, borate usually react with sugar backbone of DNA, which decreases the activity of loading drive. So we prefer buffer using TAE. We have to agarose will be dissolved in TAE. It should be mixed very well. So the solution is heated in the oven and then it have to be cooled at 55 degrees Celsius in a water bath. Next, the sample preparation. Loading buffer contains a dense compound glycerol and it includes colored dyes such as bromophenol blue. These are used to add density to samples to provide color and simplify the loading process. The sample we take in small amount, so to fill the well with its sample we use glycerol and bromophenol blue. It gives density to the sample to sink it very well in the gel and then it provide color and simplify the process of loading. Then the running buffer we are using in electrophoresis chamber is triacetate EDTA. The procedure. We have to take an horizontal. First we have to take an electrophoresis casting tray. Here two sides are left open. To that we have to cover with a tape. Then we have to pour our gel prepared, which we prepared before. We have to pour the gel by placing a comb in it. The comb, it forms the wells. That means wells. We have to leave this gel for in a normal room temperature to get solidified. Let us say 15 to 20 minutes to get solidified. After it gets solidified, we have to carefully remove this comb. Then it forms a well in this gel. And place this prepared gel carefully in the electrophoresis chamber. After placing this gel in electrophoresis chamber, fill the samples to the wells. For the first well, we have to place a DNA ladder. DNA ladder means It is used as a standard reference. Standard reference which is contains the noun, which is contains the noun length of DNA fragments. That used to compare the DNA with other fragments. Pour the samples to these on wells, then supply the electric current at the voltage 70 or 80 voltage. We have to connect this to here. We have to place a cathode and here anode. Because DNA fragments are negatively charged particles, so if we connect this means if we supply a power, the DNA particles slowly move towards the anode, which is positively charged. The smaller particles will move faster than the larger molecules. Then we have to visualize this sample in the UV light, which gives a which will look like this. These are the DNA fragments, and here are the smaller fragments, and here large fragments. This is the flow chart of the procedure. A casting tray, we are pouring the gel prepared to casting tray, and next by placing the comb we form wells. To this wells we are injecting the sample by placing the DNA ladder. We have to place DNA ladder only in the first, only in one wells to get to make comparison with other DNA fragments. And these are the DNA fragments. They move here from here, and final result is this. Applications of agarose gel electrophoresis. HEA is mainly used to separate DNA fragments based on their size. We are using this technique to separate the DNA fragments based on their size, and it helps to check whether PCR amplification was successful and if the desired DNA fragments was obtained. We are using this to check the PCR amplification. PCR amplification means polymerase chain reaction. In that we are going to produce so many copies of DNA by using a small copy of DNA. By using this process, we are going to check whether we get a desired DNA fragment. Specific DNA bands can be cut from the gel and purified for further experiments like cloning and sequencing. Here, DNA fragments, by using this process, we can easily recognize the desired position of DNA fragments to cut and purify for further experiments like cloning. Cloning means same, to copy the DNA fragments, and sequencing to order the precise nucleotides such as ACTG. AGE helps to check the integrity of isolated DNA or RNA. It will help to check the stability of DNA, DNA stability in other words, its proper function is to store the information. It is used to confirm whether restriction enzymes have properly cut the DNA to expected sites. That helps in DNA fingerprinting and identification in forensic investigations. It will help in forensics investigation to check who is the criminal. For using this process, we can use that. Next, the second type of agarose electrophoresis is SDS PAGE. The full form of SDS PAGE is sodium dodecyl sulfate polyacrylamide gel electrophoresis. They may ask for 2 months to expand SDS PAGE. Principle of SDS PAGE. SDS PAGE is a separate proteins based on their molecular weight. By using SDS PAGE, its full form is sodium dodecyl sulfate polyacrylamide gel electrophoresis. Principle of SDS PAGE. SDS PAGE is to separate proteins based on their molecular weight by using electric field and polyacrylamide gel matrix. Using this process, we are going to separate protein molecule based on their molecular weight by supplying an electric field in a polyacrylamide gel matrix. What the function of SDS, sodium dodecyl sulfate, is it is used as a detergent which dissociates protein into their individual polypeptide subunits and gives a uniform negative charge along each denatured polypeptide. Usually proteins are in different shapes of structure. It contains tertiary, secondary or quaternary. It is complex structure. By using this, it will denature the non-covalent bond and helps to form the protein into linear structure, which helps for this experiment, and it helps to give uniformity negative charge to proteins. Next, the requirement: buffer, glass plates, electrophoresis tank, foam, spacer, binder clips, and some chemicals. Protein sample, polyacrylamide gel are the requirements to perform this process. And these are the glass slides. Here we use a vertical setup. A glass, we have to take two glass plates. We have to place this glass plate in vertical position, and to pour gel in between two glass slides, we are using a spacer. Then to cover this glass, we are using a binder clip to give a tight support to the glass slides, and the sample foams to form wells. This is the final setup, a vertical setup. Here the electrophoresis chamber, and we are pouring sample to the wells. Proteins might also be treated additional with beta-mercaptoethanol. This substance is intended to dissolve the disulfide bonds which cannot be denatured by SDS. In addition to SDS, we are using beta-mercaptoethanol. It also breaks down the disulfide bond and helps to form the linear structure of proteins. Protein sample is treated with SDS and beta-mercaptoethanol, resulting in unfolded proteins, all having nearly equal charge and mass. And polyacrylamide gel electrophoresis is divided into two divisions, that is stacking gel and separating gel. The preparation of this stacking gel. Here the pH matters. The stacking gel pH is 6.8. And a separating gel pH is 8.8. We are using same components to prepare this gel, but in different composition. Acrylamide, it should be used in low concentration, and Tris HCl buffer. The pH is 6.8. It will use to maintain the pH. And SDS, sodium dodecyl sulfate, it will help to denature the proteins, unfolding bonds. And distilled water, we are using this additionally if we want to adjust the final volume of the gel. Then APS. Full form of APS is ammonium persulfate. We are using this as an initiator for polymerization. Polymerization means it is the chemical process where smaller molecules such as monomers are going to form a long chain. Here SPS is act as polymerization. By using this SPS, we will convert the gel into solid form, and it helps to start the chemical reaction with that links the acrylamide molecules to form the long chain. Then it will form the solid gel. Then TMID, its full form is N,N,N0,N tetraacetyl ethylene diamine. That's a long line. I repeat once again, tetraacetyl ethylene diamine. It will act as a catalyst for the polymerization, and it will also helps for APS and to form the free radical more faster and to initiate the polymerization. For separating gel also we using same components such as acrylamide, Tris HCl buffer, and SDS. distilled water, APS and thymic, but it's in the pH of 8.8. Then we have to prepare the protein sample. To prepare, we are taking protein as a sample. For that, this HCl, it is in the pH of 6.8. Then SDS and beta mercaptanol we are using to denature the protein structure, and glycerol to give density to the sample, to sink in the gel very well. Then bromophenol blue, it gives color. We have to boil this sample for 3 to 5 minutes to denature the proteins and then cool this sample. Next, the procedure. 2 glass slides by using a spacer in between. First, we have to pour this separating gel, then the stacking gel. After stacking gel, we have to place the comb or casting comb in it. After it starts completion of polymerization, we have to remove this comb. It forms the wells. To these wells, we have to pour the protein sample. First protein should contain protein ladder. Same thing, the protein ladder is used as a standard reference to know the length of where the proteins reach length of the proteins. All the wells are filled with sample. Then place this gel in the electrophoresis chamber. And then this is cathode. The well should be in the size of cathode size, and this is anode. Let's supply the current to this gel. Here the buffer we are taking is glycerol. The glycerol pH, it has 3 different pH. In neutral, it has 5.9. Less than 5.9, it will become positive charge, and more than 5.9, it will become negative charge. When we supplied power, here the glycerol will have, in the buffer, glycerol will have positive charge because the pH of glycerol is 6.3. When it will come to this gel, the glycerol will slightly negative because its pH is 5.9. It is slightly near to the separating gel pH, which has 6.8. The stacking pH has 6.8. So in this stacking gel, the glycerol will become slightly negative. So here the first fluorine ion will move faster than the SDS protein sample, and last the glycerol. When it reaches to the separating gel, here the pH is 8.8. The glycerol become more negative. Then here fluorine will move first, and then the glycerol, and last SDS protein samples. After the sample, before the sample reaches the bottom of the gel, we have to stop the power supply and place this gel in the Roopashree brilliant blue stain. We are using a Western brothing method. By placing this gel in the Roopashree brilliant blue, we can able to see a blue protein bands. Here are the small molecular weight proteins, and here will large molecular proteins. And this is the flow chart of this experiment. The glass slide is filled with the separating and stacking gel, and you have to pour the proteins to the wells and supply the power. The proteins will start move till the bottom of the edge. Then take the gel and visualize in this gel in the chromatic brilliant blue stain. Applications of SDS page. It will help to determine the molecular weight of the proteins. Here we are going to separate the proteins based on their molecular weight, and this will help to know the correct molecular weight of the proteins. And to check purity of protein sample, it is used to test whether a protein sample is pure or contains contaminating proteins after purification. And this sample is used to analysis of protein composition, helps identify and compare different proteins present in the cell tissue or the both in the cells and tissues or mixtures. And this process will help to verify the recombinant protein expression. That is, it helps to confirm whether a desired protein has been successfully produced in bacteria or plant expression systems. Then in biomedical and diagnostic applications, it is used in identification of disease-related proteins, and we are using this in a quality control test for vaccination and in the drug production. The next is in research in molecular biology and biochemistry. This will help to research in the field of molecular biology and biochemistry and for cloning studies and enzyme analysis. And these are some important points. You can note on these important questions. We are going to separate using HEE. Can anyone answer? The molecules we are going to separate. Then the reference notes, class notes, Wikipedia, Science Director, Genome and slide sheets. Thank you. Thank you, ma'am. Thank you all. I hope you all understand very well. If you have any doubt, please you can answer.